Hello, my name is Daryl Daniels, and today I'll be going over the last interactive case study, number 8, which I used the immunosuppressant drug tacrolimus Prograf. So basically for this assignment, we were supposed to just pick an immunosuppressant drug that was used for kidney transplants, and this was the drug that I chose. So basically I'll just be going over like what it's used or why it's used for transplant, what it is, the pathways that it targets, and just like going into further depth in the pathways. And then I just have a picture of the mechanism that I like to discuss. All right. So first, I'd like to say that as of 2021, this drug is FDA approved to use in combination with other immunosuppressant drugs that will target or prevent organ transplant rejection in adult and pediatric patients that are recovering from lung transplants. So before this, actually, when it was first approved, it was for liver transplants, then kidney transplants, and heart transplants. I actually chose this drug because I work on a hematology oncology unit in a pediatric hospital, and we actually use this drug specifically for our bone marrow transplant patients as an immunosuppressant drug. So I just found it interesting and wanted to learn more about it. So why is this drug used for transplants? This is used for transplants because when patients receive an organ transplant, the white blood cells try to get rid of or reject the transplanted organ, and this drug works by suppressing the immune system to prevent white blood cells from trying to get rid of the transplanted organ. So at my hospital we call this drug Tacro, so I'm just gonna call it Tacro for the time being. So what is Tacro? It essentially is an immunosuppressant drug that is used to prevent and treat organ rejection in people with kidney transplants. But as we just talked about, it's not just for kidney transplants. It's just an immunosuppressant drug that's used for a lot of things. So it essentially prevents rejection by inhibiting T lymphocyte cells. So here I just have the different pathways that it targets. So we have inhibition of multiple pathways, calcineurin, the C junction in terminal kinase, and P38 pathway. And it also induces increased expression of transforming growth factor beta 1. And the effect is specifically focused on the T cells. Next. So here's the first pathway I just wanted to discuss, and I have a nice picture here just to explain everything that I'm also mentioning. So inhibition of the calcineurin and NFAT. NFAT is essentially the nuclear factor activated T cells, and calcineurin is a calmodulin dependent phosphatase, and NFATs translocate to the nucleus where they associate with other transcription factors and bind to DNA to promote transcription of interleukin, which is very essential to this process. Okay, so essentially, calcineurin is stimulated during T cell activation by a chain of events involved in calcium and calmodulin. And the normal mechanism of it is to dephosphorylate NFATs. But when Tacro is used, the effect essentially is to prevent its ability by blocking its ability to dephosphorylate and activate this pathway. And by doing that, it blocks transcription, which affects the transcription of cytokines, and which is important to the immune response. So here we just have this. We have a T cell right here, and then the cytoplasm of the T cell. So essentially, you have all these things that are inhibiting this pathway from doing what it's supposed to be doing. And when this happens, it prevents the dephosphorylation of it, which allows it to not do its things and inhibits interleukin, which we'll come to find out that interleukin is essentially one of the genes for it is IL-2. All right, next is inhibition of JNK. So this essentially is the inhibition of mitogen activated protein kinase pathways, which is this right here MAPK. And MAPK is a signaling cascade that's involved in a wide variety of processes within the immune system. So here we have both of these are subgroups of MAPK. You have JNK and then P38. So once activated, they're activated via the signaling cascade by T cells and can translocate to the nucleus. They essentially promote transcription of interleukin and other cytokines. But when Tacro is put into play, it essentially prevents the transcription or expression of IL-2 gene, which is the interleukin gene, and it does this by blocking and inhibiting the pathways upstream of the MAPK pathway. And then the last thing that I had is how it essentially just induces transcription of the growth factor. So TGFB1 is this growth factor that I was talking about, transforming growth factor beta 1. So with increased expression of this, studies have shown actually that patients with end-stage renal disease who essentially need a kidney transplant, they're presented with increased growth factor beta 1 and protein expression after treatment with the drug, but it isn't it's not definitive kind of thing. So basically studies believe that the increased expression may actually be associated with nephrotoxicity and not acute rejection of the actual organ itself. So more about TGF beta 1. It is a cytokine that's important for the regulation of the immune system, and its mechanism is to essentially inhibit the interleukin gene, so IL-2 dependent T cell proliferation, and it exerts immunosuppressant effects within T cells, which is beneficial. Then here I just have more of just the general mechanism of the drug, just a breakdown of the different pathways that I mentioned. So Tacro essentially binds to FK binding proteins, and for this specifically, it's this one right here. And then the MAPK8 is inhibited via the JNK and P38 inhibition, which takes place more in the nucleus because it has to do with transcription. And then it'll inhibit interleukin transcription, and it exerts its immune response by being an immunosuppressive drug. All right, and then I just have my references, and that's all. Thank you.